if we take care of nature.
The opportunity to rewrite life at our fingertips, and just like nuclear power,
life wants to flourish. DNA wants to go forward.
Nobody knew what to expect with genetic engineering, but they pressed the button.
We need to be part of that.
The product of late 20th century genetic engineering.
Will take care of us.
Hello all, my name is Paige, and I'm excited to share with you a little bit of research into genetic engineering that I've been doing over the past few weeks. You don't need me to tell you that we are living in crazy times that are both exciting and terrifying. However, it is only a matter of time before news and social media blows up with the next breakthrough into gene editing technology.
Case in point, the 2010s already has a bunch of genetic engineering related memes.
It is something that has captured our imaginations in the past, and I don't think it's going away anytime soon. If anything, I think it will come back stronger. In order to understand the hubbub, let's take a look into the history of gene editing. DNA, or deoxyribonucleic acid, that's a long word, is a molecule that carries the genetic instructions for the development, functioning, growth, and reproduction of all known living organisms and many viruses. It consists of two long strands that coil around each other to form the famous double helix, which you've probably all seen before. Each strand is made up of smaller molecules called nucleotides. These nucleotides contain a sugar, a phosphate group, and one of 4 nitrogenous bases. These are adenine, cytosine, guanine, or thymine. The sequences of these bases along the DNA strand encodes genetic information with specific sequences representing genes that determine an organism's traits. RNA, ribonucleic acid, is another important molecule. However, unlike DNA, uracil replaces thymine to pair with adenine. It is also single-stranded. While DNA stores genetic information, RNA plays various roles in protein synthesis and gene regulation. The Human Genome Project began in 1990. It consisted of a team of scientists who sought to sequence the entire human genome. After 13 years and the cost of over $3 billion, they were able to achieve this amazing feat 2 years ahead of schedule. Now, the first instance of gene therapy was published in 1962, where recombinant DNA technology was developed. The first production of recombinant DNA using restriction enzymes didn't occur until the early 1970s, however. Recombinant DNA technology entails the merging of DNA from distinct species, followed by the introduction of the hybrid DNA into a host cell. Host cell is frequently a bacterium. Scientists at UC San Francisco and Stanford employed restriction enzymes to precisely cleave DNA from various species at specific locations. Subsequently, they combined the cleaved strands from the different species. Shortly thereafter, viral vectors began to be used in gene editing. Viruses have the ability to splice new genetic information into DNA of bacterial cells. Scientists theorized that we could make the viruses deliver desired genetic information into test cells. While viral vector have not always been successful, this method, the idea of using viruses as gene delivery vehicles has played a huge part into genetic research. Those first two methods are examples of gene therapy, which randomly adds therapeutic genes into the host genome. This caused concerns by how little control was had over the edits being made, and these concerns gave rise to gene editing, which allows us to have more control by using nucleases to intentionally cut out pieces of DNA and introduce those changes. Some examples of gene editing include ZFNs, TALENs, and CRISPR or Cas. For the sake of time, I'm only going to explain a little bit about CRISPR technologies, which is by far the most well-known and the simplest method. CRISPR technology is a revolutionary tool used for editing the genome. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. These are segments of DNA that are found in the genomes of bacteria and other microorganisms. The CRISPR-Cas9 system works like molecular scissors, allowing researchers to precisely modify genes within an organism's DNA. The Cas9 enzyme, guided by RNA molecules that are designed to match a specific sequence within that genome, can cut out DNA at that location. Once that DNA has been cut out, We are then able to insert, remove, or alter those specific sections of the genetic code, leading to changes in the gene function. If you would like to learn more about the development and discussion around CRISPR, I highly recommend reading A Crack in Creation, Gene Editing and the Unthinkable Power to Control Evolution by Jennifer A. Doudna and Samuel H. Sternberg. They were both some of the first scientists to work with CRISPR, and their book has been highly educational and has given me a lot of the information that I'm using in this video. Wait a minute, that's not quite what I mean. When I say the death of nature, I don't mean that nature is literally dead. Rather, I am referring to the book under the same name by Carolyn Merchant. Carolyn is an ecofeminist, philosopher, and historian of science. She discussed how prior to the scientific revolution in the 16th and 17th centuries, nature as a whole was viewed as living. It was the feminine Mother Nature who provided life to all, but varied between nurturing with abundant resources to unpredictable and cruel with fierce storms and catastrophes. She could flip-flop between both. Ancient people sought to live within her approval and saw themselves as being largely dependent on her and her goodness. As time passed, however, and led up to the scientific revolution, early Enlightenment thinkers such as Sir Francis Bacon began to teach that in order to gain new knowledge, nature needed to be forced to reveal her secrets. He used very problematic language, which we definitely do not use now. In his mind, nature could be pressed to give up her resources and knowledge in service of mankind. A huge part of Carolyn Merchant's thesis is that the scientific and intellectual developments of this time not only led to economic and intellectual progress, but also to social repression, specifically of women. This aspect of her work is hugely interesting. Definitely go read her works, but just for the sake of this discussion, and for the sake of time, frankly, I'm just going to focus on the idea of nature as seen as a living thing in general. Or not living. Let me explain. Because of Isaac Newton and his new physics, it was possible to describe the visible world using universal laws. These laws could be used to answer questions that people have had about the natural world since ancient times, and they were very logical. The combination of these natural laws, along with the recent development of the pocket watch, led to the watchmaker theory. As described by William Paley in 1802, anyone finding a pocket watch in a field will recognize that it was designed intelligently. Living beings are similarly complex and must be the work of an intelligent designer. This is obviously a theological theory, which we're not going to discuss, but it is relevant because of the surge of deists at the time who believed that God was the intelligent watchmaker, and they believed that nature was not an organism, but a machine, much like a watch. Instead of being alive and conscious, nature was now an inanimate object. The death of nature. It is here we see the shift from humanity being dependent on nature and its changes to instead taking control of it. We now view the world as being constructed from atoms, and while we know that there's a lot more than just Newtonian physics guiding the movement of the universe, we've gotten to the point that our new discoveries work for us. We have been making advancements in medicine, transportation, communications at astonishing rates. Most of these advancements come from us using the world and nature around us and altering it and controlling it to achieve a specific desired outcome. Speaking of controlling nature, this is where we can tie back into gene editing. Genetic engineering is a perfect example of humankind taking control over nature. Our genetic codes are produced by millions of years of evolution. We have recently tapped into the ability to be able to alter them. There are infinite stances that you can take on gene editing and whether it's acceptable or not. I'll just break it down into a few pros and cons. On one hand, genetic engineering can be hugely valuable in the treatment of diseases. One example is that of sickle cell disease, which is caused by a mutation in the hemoglobin beta gene. If someone receives 2 copies of this recessive trait, then they suffer from high levels of pain, fatigue, and other symptoms. Current treatments involve blood transfusions, bone marrow transplant, and the drug hydroxyurea. Hope I pronounced that right. A much more efficient way to treat the disease, however, would be to target the mutation itself. For people who have similar kinds of illnesses, the option of being able to treat the disease at the source would be a huge relief. There have also been plans to genetically modify mosquitoes in order to prevent them from spreading malaria, which is another one of the most deadly diseases on the planet. These are just a few examples, but that's kind of like one of the big ones, is like treating disease. When it comes to the food industry, as far as genetic engineering has been concerned, people have been genetically modifying our food to some extent for hundreds of years. Crops and livestock have been selectively bred to have certain traits as long as people have raised them. What is breeding if not less controlled method of genetic manipulation? Genetically engineering plants has the ability to improve crop yields and nutritional content, helping to address global food security issues. Genetic engineering can also be used to develop crops that are more resilient to pests, diseases, environmental stresses, and reducing the need for chemical pesticides and herbicides, which we know are harmful for the environment. And speaking of the environment, genetic engineering has applications in environmental conservation, such as developing organisms that can clean up pollution or restore damaged ecosystems. One really good example is that of a bacteria that has been developed to help clean up after oil spills. It literally eats the oil. Scientists are also working on developing different kinds of bacteria or fungi that can help break down plastics, which normally take thousands and thousands of years to go away. For those who claim that gene editing is unnatural and that we have been specifically evolved to be how we are today, Jennifer Doudna points out, it turns out that nature is less an engineer than a tinkerer, and a fairly sloppy one at that. Its carelessness can seem like outright cruelty for those people unlucky enough to inherit genetic mutations that turned out to be suboptimal. Similarly, the argument that germline editing is somehow unnatural doesn't carry much weight with me anymore. When it comes to human affairs, and especially the world of medicine, the line between natural and unnatural blurs to the point of disappearing. We wouldn't call a coral reef unnatural, but we might use the term for a megalopolis like Tokyo. Is this because one is crafted by humans and the other isn't? In my mind, the distinction between natural and unnatural is a false dichotomy, and if it prevents us from alleviating human suffering, it's also a dangerous one.
Well, she was determined to prove that genetic power could save lives.
On the other hand, genetic engineering has its fair share of environmental concerns. One major concern is the potential for unintended consequences, such as the creation of new allergens or toxins in genetically modified organisms. There are also worries about the impact of these organisms on their ecosystems, including the potential for them to outcompete with native species or disrupt delicate ecological balances. While there may be one scientist who merely seeks to remove the ability of mosquitoes to spread malaria, there are others who would wish to just completely eliminate mosquitoes, which would have absolutely far-reaching, devastating effects on the rest of the food chain. Additionally, the use of genetic engineering on humans raises very complex ethical questions, such as the potential for creating designer babies or widening the gap between the rich and the poor if genetic enhancements are available only to the wealthy. It could become possible that social classes are determined not only by wealth, but also by genetic makeup. This scenario is explored in the 1997 film Gattaca. This movie, it's one of my all-time favorites. Like, if you haven't seen it, please do. It stars Ethan Hawke as Vincent, a genetically inferior man, also known in the film as a degenerate, who assumes the identity of someone genetically superior in order to achieve his goal of going to space. Along with discussions on genetics and class, the movie also gives commentary on how much of our personal DNA is considered private or public information. Even Jennifer Doudna herself has some reservations about fully jumping into the acceptance of genetic engineering. Being an intellectual, she knows the awful history of eugenics and those who sought to do harm to groups of people. She writes about a nightmare she once had, saying, "A colleague approached me and asked if I would be willing to teach somebody how gene editing worked. I followed my colleague into a room to meet this person and was shocked to see Adolf Hitler in the flesh seated in front of me. Fixing his eyes on me with keen interest, he said, "I want to understand the uses and implications of this amazing technology you've developed." His terrifying appearance and sinister request were enough to jolt me awake. As I lay in the dark, my heart racing, I couldn't escape the awful premonition with which the dream had left me. The ability to refashion the human genome was truly an incredible power, one that could be devastating if it fell into the wrong hands. The thought frightened me even more because, by this point, CRISPR had been widely disseminated to users around the globe. Is our newfound abilities to rewrite genetic code the final nail in nature's coffin? Are we getting ahead of ourselves and demanding control over the natural world? Or are we learning how to better survive and adapt to our surroundings? I don't think there's any one correct answer. Just as of now we question the ethics of using gene editing, there may one day come a day that we question if it's moral not to. I'll end by saying that I don't think that there is any one clear-cut stance. My hope is that you will take this little information that I have shared with you and go and do more research for yourself. This is only like the tip of the iceberg, both into gene editing as well as into the ideas of the death of nature. Form your own opinions. Because whether we like it or not, genetic engineering is a part of our present and our future. Regulatory frameworks and public acceptance are crucial in ensuring that genetic engineering is used responsibly and for the greater good. Thank you for sticking around, watching until the end. I'll link some of the resources and related articles in the description below. Bye!